Good morning and welcome, everyone. We'll just give it a couple of minutes and we will start our presentation. Okay, I think it's safe to begin. Hello, everyone, and welcome to today's webinar. I'm Marissa Fraser Morera with Pion, and I will be moderating today's event. This webinar is titled Flavonoids, an example of challenging pKa determination, and will be presented by Dr. Rebecca Reese. Rebecca is the principal scientist at Pion, where she has the overall responsibility to design and oversee research experiments, deciding which projects should be prioritized, and present the results and conclusions at conferences and to international scientific publications. Rebecca obtained a European PhD in analytical chemistry from the University of Barcelona in Spain, and her thesis and master's were focused mainly on pKa, ionization constant, analysis, and studying and evaluating a diversity of techniques, such as potentiometric and spectrophotometric instrumentation like HPLC, CE spectrometers, and potentiometers, media, and different calculation approaches for the determination of this property. Lipophilicity, log PD, and solubility also form part of her doctorate, and Rebecca publishes her work in internationally recognized scientific journals. Rebecca worked at the pharmaceutical company Almirall AS as a senior scientist and also as an assistant professor at the University of Barcelona, obtaining a valuable understanding of both worlds, big pharma and academia. Her expertise in physical-chemical properties has been expanded over the years, increasing her knowledge in dissolution, permeability, absorption, and the study of the effect of different families of excipients on the physical-chemical properties and their role in formulations. Now, let's go over to some housekeeping items. You are encouraged to contribute to the success of this webinar by asking questions at any time during the presentation through the Q&A box on your screen. Our speaker will answer them at the end of the presentation, and as your moderator, I will be posting as many as time permits. And any questions that we don't get to today will be answered by email after the event. Now, if you need technical assistance during the webinar, please type your query into the chat box. And also, a recorded version of this webinar will be available on our website within about 24 hours. And you will also receive an email with a link to the recording. This is sent to all that register for today's event. Now, without further ado, let's get ready, and I will hand things over to Rebecca.
Thank you, Marissa, for your introduction, and welcome to all of you who have joined us today for this webinar on flavonoids, an example of challenging pKa determination. My name is Rebecca Ruiz, and I'm the principal scientist at Pion. This presentation is divided into 3 parts. In the first part, I will talk about the ionization constant, the importance, and a quick explanation of the basics of the pKa. I will also talk about the instrumentation used for the pKa determination in this work, which is the Pion Sirius T3. In the second part of this webinar, I will expose the case study of the flavonoids, the importance of these compounds, and the difficulties found on the determination of their pKa's. For that, I will focus on 2 flavonoids of the 9 studied, quercetin and fisetin. In the third part, I will play a video about the use of the Pion Sirius T3, which will demonstrate the determination of the pKa using the fast UV technique. The pKa value of a compound not only provides an idea of the degree of ionization in the given medium, but also influences other properties of the compound, such as absorption, solubility, lipophilicity, etc. Each of these properties shown in this slide are affected by the pKa, since the behavior of a compound is different when it's ionic or neutral. For example, a compound is more soluble in water when it's ionic, but is more lipophilic when it's neutral. That is why obtaining this parameter is important in ADMET processes. For example, if a sample has 1 pKa, we can say that the pKa is the pH at which half the molecules in aqueous solution exist in ionized form and half exist in their neutral form. So it's the pH when the 50% of the molecules are ionized and 50% are neutral. As a way to understand the ionization constant, I always like to use the example of thymol blue. As this compound is an indicator, then every species involved in a pKa has a different color. Thymol blue is an acid and has 2 pKa's. The first one is 1.7 and the second 8.9. If the solution is a low pH, below the first pKa, the sample is neutral and is red. If we increase the pH between 2.8 and 8, the molecule loses its proton, marked with a red circle. The main species is monoanionic with 1 negative charge, and the color of this species is yellow. Increasing the pH higher than the second pKa, 8.9, the sample loses the second proton, marked with an orange circle. The main species is now dianionic with 2 negative charges, and the color of this species is blue. Many drugs are ionizable molecules, and so they have pKa values. Ionizable drugs have structural groups that pick up or lose protons, and they convert between different ionized forms as the pH changes. The importance of knowing which species will be in solution at the target pH will be done by the pKa. Moreover, the neutral species, our red species in the example of the thymol blue, will be able to penetrate and diffuse through the biological membranes of the target organ. We developed the Pion Sirius T3, an instrument for the determination of the physicochemical properties, such as ionization constant, pKa, lipophilicity, log P, and solubility. The instrument is a fully automated potentiometer, which uses 2 techniques for the determination of the pKa, the spectrometric for UV-active compounds and the potentiometric, which I will explain in more detail in the next slides. Sirius T3 has a measurement cell of just 4 milliliters, in which a mini pH electrode of 3 millimeters in diameter measures the millivolts at every single experimental point, as well as a capillary set encased in a ceramic tube of 2 millimeters diameter, which dispenses the titrants and media. The cell also incorporates a stirrer, temperature probe, and a spectroscopic UV probe, which detects absorbance and turbidity. This is all immersed in just 1 centimeter diameter glass vial, enabling titrations in just 1.5 milliliters of solution under argon. Additionally, the instrument detects turbidity using a red LED that shines through the solution onto a photodiode detector. As I have already mentioned, the Sirius T3 determines pKa's using 2 techniques, the spectrometric and the potentiometric. The spectrometric technique is for UV-active drugs, meaning compounds with chromophore groups close to the ionized center. This technique calculates the results from the changes in the UV spectra as the sample is ionized with the change of the pH, based on the theory that the two species involved in the pKa have different spectrum. For the spectrometric technique, we have two methods, the fast UV and the UV metric. The fast UV method is a screening method because it only takes 6 minutes per titration, obtaining a result in 20 minutes for triple assays. It operates between pH 2 and 12 and uses a safe reference spectrum, so it's quick and efficient. The second spectrometric technique is the UV metric method, which operates over a wider pH range, below pH 2 and above pH 12. And it uses a fresh reference spectrum for each sample. The UV metric technique is therefore more precise than fast UV, but requires a longer assay time to get a result. Both methods use a small quantity of sample, just 5 microliters of DMSO stock solution of 10 millimolar, or 0.02 milligrams. Both determine pKa's in aqueous and cosolvent media, in the case of poorly soluble samples. For the determination of the flavonoids pKa, we were using both techniques, fast UV and UV metric, and I would like to explain in the next 2 slides how these techniques work. At the beginning of the assay, a reference is needed for any of the two techniques, as the media absorbs. The media not only has the water solvents and the titrants, but also has the DMSO or the solvent used in the sample stock solution. Once the reference spectrum is subtracted from the sample spectrum, the sample absorbance spectrum is obtained. For a pKa determination, both species involved in the ionization need to have different spectrum, and enough different to be able to measure the changes of the absorbance at every wavelength between 200 and 700 nanometers when the pH changes. In this example, I have a base, and looking at the graph absorbance versus wavelength, the neutral species in blue absorb less than the protonated species in red at the wavelength of 300. In the second graph, we have the absorbance versus pH at the wavelength 300 nanometers. The neutral species will be in solution at high pH, and the protonated at low pH. But at 300 nanometers, the neutral species will absorb less than the protonated. This is the reason of the change in the absorbance with pH. Selecting another wavelength, for example at 275, we are in the opposite case, where the neutral is absorbing more than the protonated. As the neutral will be in solution at high pH, the change in absorbance will be from high to low absorbance when decreasing the pH. The software performs this exercise with all the wavelengths, obtaining the following spectra, which it will determine the pKa at the absorbance change, where 50% of the sample will be ionized and 50% neutral. The instrument detects precipitation by UV, as well as using the turbidimeter. The sample needs to be completely dissolved at the beginning of the assay, and so the absorbance will be fixed at 0. When the titration starts, the absorbance will be recorded during the assay following one wavelength, 500 nanometers, where it is assumed that the sample doesn't absorb. If the sample precipitates, the absorbance will increase because of the scattering occurred by the particles in solution. If the increase of the absorbance is higher than the threshold assigned for the technique, the software will paint a yellow area to show where the precipitation was detected. If this happens, the assay needs to be repeated using a co-solvent media if water was used, or a higher percentage of the co-solvent used. The potentiometric technique is for UV-active and non-UV-active drugs, in which the pKa is obtained by titrating the sample between pH 2 and 12 and calculating the pKa using mass and charge balance equations. The pH metric technique is the most powerful technique because it can find any pKa regardless of whether or not the sample is UV active. However, it requires a greater amount of sample and the probability of precipitation is higher. This slide shows the validation of the pKa techniques used by T3, the UV metric versus the potentiometric. You can also find on this slide interesting published papers about the techniques covered. Case study: flavonoids pKa determination. Flavonoids are polyphenolic compounds which are starting to emerge as a potential new class of drugs due to their extent of pharmacological activity. They play multiple roles in biological processes, especially related to their antioxidant efficiency. Even though there is a great interest in the flavonoids in recent years, there is a little data related with their physical-chemical properties due to their molecular complexity. For instance, flavonoids show numerous phenolic groups, from 1 up to 5 or even 6, with close pKa values. Many flavonoids are not stable at basic pH, and they decompose, making the determination of these high pKa's quite difficult. And finally, they are poorly soluble in aqueous media, which complicates even more the experimental determination of their physicochemical parameters. This study is published, and the pKa results of the 9 flavonoids shown in this slide were determined, but for the purpose of this webinar, I will be focused in only 2 of them: quercetin with 5 high acidic pKa's and fisetin with 4 high acidic pKa's. This slide shows the pKa's of the 9 flavonoids found in different publications using different techniques. Please note that the last 2 values are obtained using the predicted packages Spark and Percepta. It's quite surprising the lack of integrity in the data found in literature. For the 5 pKa's of quercetin, only 1 publication was found, which determined the 5 of them. However, all 5 pKa's have lower values than the rest of the published pKa's, with differences between 1 and 2 log units. For fisetin is even worse, as no literature was found with all the 4 pKa's published. The difference between the pKa's found are even higher than 1 log unit. Let's start with quercetin. At the first instance, for the determination of the 5 acidic pKa's of quercetin, the UV metric technique was selected, performing the assay in aqueous media. In order to avoid the decomposition of quercetin at high pH, the titration was carried out from low to high pH. However, as I have previously commented, quercetin is a poorly soluble compound and precipitation was detected at low pH, which is marked with the yellow area, making it not possible the determination of the lower pKa. When the titration reached high pHs, the sample solubilized, but showed signs of decomposition as the UV signal at high pHs cannot be refined. Overall, only 3 pKa's of the 5 could be determined with this technique. In order to study the extent of the decomposition, 2 titrations were performed using the UV metric technique in aqueous media, titrating from opposite directions, from low to high and high to low. It's interesting to observe the difference in the shape of the spectra for the different species involved on the pKa's, where the decomposition is affecting mostly the second titration. Titrating from high to low, as expected, the precipitation at low pH has not been observed. There is an additional pKa at low pH, which is not observed from low to high. For both titrations at high pH is observed the effect of the decomposition as the data cannot be refined. Overall, only 4 pKa's could be determined for both titrations, and they are not even in good agreement. As we couldn't determine the pKa's of quercetin using the UV metric technique, the fast UV in aqueous media was then used. The fast UV is also a spectrometric technique, however, titrations can be performed in just 6 minutes. So in some cases, fast UV is quick enough to avoid precipitation or even decomposition. And it's exactly what happened here. As it shows in this slide, precipitation didn't happen at low pH, and the decomposition wasn't observed at high pH. Again, in order to study the extent of the decomposition, 2 titrations were performed, using the fast UV in aqueous media, titrating from opposite directions. The spectra of the different species involved with the pKa's are completely different for both titrations, as the decomposition happens when titrating from high to low. Titrating from high to low, there is an additional pKa at low pH, which is not observed from low to high. And at high pH is observed the effect of the decomposition as the data cannot be refined. Finally, 5 pKa's could be determined with the fast UV performing the assay from low to high. We have observed that using the fast UV, the composition at high pH was not observed when titrating from low to high pH, as the titration is quick enough to avoid degradation of the sample. Then it would be interesting to know at which pH the composition started. Using the UV metric technique, we have found that degradation of flavonoids happens above pH 9.5. But what about using the fast UV? Therefore, several titrations were carried out from high to low, starting at different pH values to be able to find the pH where the decomposition did not happen. For that, there are 2 decomposition effects to follow: the low pKa, which appears as a result of the decomposition, and the spectra shape change associated to each species involved in the pKa, which can be compared with the titrations from low to high. In this slide, it's easy to observe that from the titration starting at pH 12 and the one at pH 11, the low pKa result of the composition appears, but from pH 10.5 and pH 10, this pKa is not there anymore, and the spectra related with the titrations at 10.5 and 10 are similar. Then, using the fast UV, quercetin decomposed above pH 10.5. In this slide is shown the effect of the decomposition for quercetin. The graphs absorbance versus wavelengths were compared to observe the change in the spectra with the pH, when the titrations were performed from low to high and high to low. From these two graphs, we can obtain the spectra of the species involved for each pKa and how the shape of the MEX, the molar extinction coefficient, changes due to the decomposition involved. Therefore, the solid lines are from the titrations carried out from low to high, and the dot lines from high to low. Let's talk about fisetin. Fisetin has 4 acidic high pKa's, and the graphs shown in this slide are titrations performed using the fast UV at opposite directions in order to study the extent of the decomposition, as it was done with quercetin. The spectra of the different species involved with the pKa's are completely different for both titrations, as the composition happened mainly in the second titration. Titrating from high to low, there is an additional pKa at low pH, which is not observed from low to high. And at high pH is observed the effect of the decomposition as the data cannot be refined. 4 pKa's could be determined with the fast UV performing the assay from low to high. In the same way that it was performed with quercetin, we have studied the degradation of fisetin, performing several titrations from high to low, starting at different pH values to be able to find the pH where the decomposition did not happen. For that, just remember that we have two decomposition effects: the low pKa, which appears as a result of decomposition, and the spectra shape change associated to each species involved with the pKa's, which can be compared with the titrations from low to high. In this slide, it's easy to observe that from the titration starting at pH 12 and the one at pH 11.5, the low pKa result of the composition appears, and from pH 11, this pKa is not observed anymore. The spectra related with the titrations 10.5 and 10 are similar. Then, using the fast UV, fisetin decomposed from pH 11. For the case of fisetin, the graphs absorbance versus wavelength were compared to observe the change in the spectra with the pH when the titrations were performed from low to high and high to low. From these two graphs, we can also obtain the spectra of the species involved for each pKa and how the shape of these makes change due to the decomposition involved. Then the solid lines are from the titrations carried out from low to high, and the dot lines from high to low. Finally, we have published the values obtained for quercetin and fisetin, as the rest of the flavonoids studied. It's easy to understand the discrepancy between the data found published due to the difficulty in the determination of the pKa's of these samples. However, we are very proud to publish for the first time two new pKa's, the highest pKa's for fisetin and quercetin. Acidity or basicity is one of the most important properties of a molecule, since the ionization state governs other physicochemical properties such as lipophilicity, solubility, and the ADMET processes. Flavonoids are challenging compounds for the determination of their pKa's due to the poor solubility, observed decomposition at high pH, and close pKa's. Performing two titrations at opposite directions allows stability evaluation and the correct determination of the pKa's. Fast UV was proved to be a technique which avoids in this study the composition and precipitation in comparison with the UV metric technique. UV metric technique showed the composition of flavonoids above pH 9.5, when fast UV the composition was detected above pH 10.5 for quercetin and pH 11 for fisetin. Sirius T3 determines pKa's as well as log P and solubility checksum, parameters which are recognized as setting the industry standards. Several techniques and different assay settings must be used to be able to confirm results, as different techniques are complementary, giving enough information for a better understanding of the behavior of the drug. For the first time, two new ionization constants have been determined for quercetin and fisetin. I would like to thank the chemists in Pion Analytical Service, especially to Andy Kennedy and Daniel Badry. Also, thanks to Sam Lee and Imogen Knight. The University of Barcelona, to the doctors Clara Raffles, Elisabeth Bosch, Elisabeth Fouguet, and Mary-Jane Magnet. Thank you so much for attending this presentation. However, before the end of this webinar, I would like to show a quick video about the instrument used for this work, the Sirius T3, and the technique, the fast UV. Dipyridamole was selected for the experiment. Thank you. In this video, I will be demonstrating the determination of dipyridamole pKa, a basic compound using Pion Sirius T3. Sirius T3 is a fully automated system which determines the pKa values using two techniques, the spectrometric with the fast UV and UV metric, and for non-UV active compounds, the potentiometric. It has three modules, the dispenser module, the titrator, and the autoloader. The dispenser unit automatically dispenses the water, the titrants, base, and acid, neutral linear buffer for fast UV assays, partition solvent for log P measurements, and cosolvents from a 6-wave valve, which allows the addition of up to 6 different cosolvents. All these dispensers are connected with the reservoir bottles for each reagents and titrants following a color code: red for the acid, blue for the base, white for the water, yellow for the octanol, orange for the buffer, and black for the 6 cosolvents situated in the back of the tray. To avoid the contamination of the carbon dioxide, the titrants are under inert atmosphere conditions. From a bottle full of deionized water, the argon is bubbling. The wet argon will go to the water, and from the water to the base, from the base to the control bubbler. The T3 probe arms perform titrations with a high degree of accuracy through autonomous reagent addition and data collection. Assay conditions are tightly controlled and data is collected in situ and in real time using a mini electrode for millivert readings, the temperature sensor, and the UV probe connected to the spectrometer. The stirrer on the left, 6 capillaries from which reagents and solvents are dispensed, and an inert gas line which permits the titrations be performed under inert atmosphere. All these devices work in tandem to carry out precise and reproducible assays. In the titrator module, at the front is the titration and reference positions, the water wash position and the drain, 2 wash positions for the surfactant and the solvent, pH 7 buffer, and the storage solution. The titrator arm moves through these static solutions to clean the probes after and before each assay, and to calibrate the electrode. We also have an automated sonicator to help dissolution of solid samples, and a sponge for removing excess of water from vials after sonication. The autoloader module fitted 4 trays and includes the vial arm, which moves vials between the racks and the assay positions. Each vial rack has 8 rows and 6 columns, a total of 192 vial positions. Using the fast UV assay, which is 6 minutes per assay, so 20 minutes per triple titration, allows you to perform in one day 72 assays. For samples poorly soluble, the sonicator could be added automatically at the beginning of the assay to help dissolution before the assay starts. Let's switch to the instrument control screen. I will now load the templates into the well positions. In position A1, I create a fast UV buffer calibration, and in position A2, I will create a fast UV pKa assay. As a basic compound is being titrated, the direction of the titration is run from low to high pH. For compounds we do exhibit solubility issues, we can adjust the cosolvent conditions of each titration using the cosolvent volumes dialog box. Poor solubility can be compensated for either by increasing the percentage of cosolvent in each titration or by changing the cosolvent type. Then I will specify the details of the sample by creating a sample model. First, I specify the sample form, 10 millimolar DMSO stock solution, and then specify the volume. Then the sample name. This is the most basic level of information which is required to run a pKa assay on T3. However, if more information is entered into the sample model, providing at least estimated pKa values facilitate the software's auto-refinement feature. To take advantage of this, I will add a predetermined sample structure to this model, generated by the assay expert function built into T3. Assay expert is capable of producing in silico estimations of the pKa values of compounds, in addition to solubility and log-P. These predictions are tied to the structure file by assay expert and by importing the structure to the sample model. The software automatically fills out all the predicted PhysChem information into the model. Upon starting the assay, the robotics begin to move. The autoloader checks that the titration position is clear before moving the assay vial into position. And the probe arm washes the probes in the wash position. It will also record a reference millivolt readings for the electrode in the pH-7 buffer. The dispenser unit prepares the reagents needed for the assay. The probe will rinse the excess of drops from the reagents and finally will move to the sample position. It's now when the dispenser unit adds the media, aqueous or cosolvent, depending on the type of the assay. And the titrants, always starting the assay at the pH where the compound is ionized to help the dissolution of the sample, adding base when the compound is an acid and adding acid when the compound is a base. In the fast UV assay, the instrument automatically adds the water, co-solvent, and buffer reagents. Then the temperature is fixed to the value set in the assay design, in this instance 25 degrees. The medium is then adjusted to the starting pH value of 2 using the acid titron. From the starting pH, the instrument adds base, and after each addition, record the pH value of the medium at that point, the UV spectrum, and the temperature. In this view, we can see the status of the titration, including the pH reading, the temperature, and the reagent volumes added up to that point. The instrument log on the right side records all instrument actions during every assay performed. We can also view the spectral data being collected in real time. We'll skip ahead to completion. When the assay finalizes, the probe arm washes the probes in the wash positions. The T3 returns the save vial to the source position, and the robotics move back to the standby positions. The assay is safe and refined automatically by the software. The T3 refinement software visualizes the titration data as you see here, in a plot of the UV absorbance per monitor wavelength, represented by the purple lines, versus pH. We can see quite clear that around pH 5.7 there are apparent changes in absorbance over multiple wavelengths, visualized by the crossing purple lines, which indicates there is UV-active ionization occurring in this pH region. Overlaid on this plot, you can also see a distribution of a species profile, which describes the ionization of the sample according to the number of pKa which are defined in the sample model, and the current result of the pKa calculations display with these flags. The different colors of the profile represents the different ionizable states. In this case, the blue represents the cationic form and the red the neutral form. The percentage of each ionized species can be read from the secondary y-axis relative to any pH. If there is a disagreement between the model applied to the data and the data itself, then the applied model is not an adequate description of the data which has been collected. If the percentage species data points align perfectly with the model, then the model matches with the true number of pKa's within the data, and the pKa values calculated by the software can be taken to be the correct pKa values of the sample. Once the pKa calculation is complete, we can view a collection of other useful information, including the molar absorption profile of each of the ionizable states. When all the titration are processed, we can also view the overall pKa result at the top of the file, which in the case of the co-solvent pKa assays will present the aqueous pKa value extrapolated from the PhKa values determined under each of the different co-solvent conditions of the assay, using the Yasuda-Shedlocksky extrapolation equation. The individual titration results are presented in the top right. From this final pKa result, we can also view the distribution of species under aqueous conditions, from which we can calculate the percentage of each species present at particular pH values. The instrument also offers the UV metric assay, more accurate than the screening fast UV. And when the sample is not UV active, the potentiometric technique will be applied. Sirius T3 can also determine lipophilicity log P with octanol as the default partition solvent, and also solubility with aqueous or co-solvent media using the patented check-sol method developed by Pion. This video concludes here. If you have any questions or you need any more information, please contact Pion.
Hey, we're back.
Oops.
Where are you, Rebecca? There she is.
Here I am.
We do have some questions from the audience. Let's begin. And as I said, if we don't get to your question today during the live session, we will definitely reply to you via email. So let's start with the first question. Why wasn't a co-solvent used when precipitation was observed in water for quercetin?
Yeah, you are right. When precipitation is detected during an experiment, especially if the experiment was performed in water, like in our case, yes, needs to be repeated using a co-solvent because sample needs to be in solution, needs to be dissolved for the whole duration of the assay. However, in this case, we didn't use a co-solvent with these samples because they have high pKa's and they are acids. So usually the co-solvents when we are adding a co-solvent in an assay, for example, methanol water, the pKa is going to shift to a higher pH if we have an acid, and will shift to a lower pH when it's a base. So if we are using more and more percentage of solvent, then the pKa will shift more and more. So in our case, using a co-solvent, if we use a co-solvent, the thing that happens is that the pKa cannot be determined because we'll shift outside the pH limit of the technique. So this is the reason that we have to change the technique to be able to determine these pKa's, because quercetin and fisetin, they have the high pKa's. Let me check. They are around the higher pKa's. They are 11 and 12. So that means that the pKa's will shift outside of the pH limit of the technique, so not be able to measure. Hope I have answered the question. Oh, I cannot listen to you, Marisa.
I muted myself so I wouldn't interfere with you answering questions. Sorry about that. I was just saying we have another question here for pKa analysis. What is the minimum distance between two pKa's from them to still be determined with this instrument?
Yes, good question. The theory says that for the determination of a pKa, it needs 2 pH units as minimum distance from both sides of the pKa, so like a plus minus 2 pH units. But for the spectrometric technique, it depends a lot on the sample. It depends a lot on how strong is going to be the UV changes, and I mean how different are going to be the spectra of the two species involved in the pKa, and yes, and if the sample has enough absorbance. So for example, in the case of the flavonoids that I talked today, the difference between the two pKa's in all cases, they were less than 2 units. So if this happened, then to confirm that your pKa is the true one, you have to repeat the assay like at least 4 times to have replicates and confirm the pKa. And actually thinking about it, yes, quercetin and fisetin, the difference between the pKa's, they are less, even 1 unit, 1 log unit. Hope I have answered the question.
Okay, next question. If I have a sample which decomposes and I run the assays from low to high pH and high to low pH, how can I tell which pH caused the decomposition?
Yeah, this is a very good question. As yes, it's quite difficult to know at which pH the sample is decomposing. Yes, it's true that you will have different pKa results from the 2 titrations, from high to low and low to high. And yes, the spectra from the different species will be different. So yes, you know that you have the composition, but you don't know at which conditions. The quick solution for this case is having a predicted pKa, so for any predicted packages, because if you have obtained different pKa values, the predictions maybe could help to you to decide which is the pH which is causing the decomposition. And what could happen as well is that maybe the prediction said that your pKa is an acid, but you actually have a base. So something that is not matching your predictions. So this is actually another reason why the assay expert software, the software that I was mentioning in the video, is highly recommended to have it, especially if you don't have access for any predicted pKa's. But yes, let's say that you don't have a predicted package, not predicted pKa's, what you can do. So the best that you can do is to use all the techniques available in the instrument, performing the titrations from opposite directions. Then you need to compare the different results between all the techniques and a little bit of investigate from which pH the decomposition is happening. So the reason of this is because there are techniques that take long time to finish the assay than the others. For example, potentiometric technique needs around 25 and 30 minutes to complete the titration. Then the composition will happen earlier, let's say it. And fast UV needs only 6 minutes so the per titration, so the sample will decompose like later. So, okay, what I try to say is if your sample, for example, decompose from high to low, and you use a potentiometric technique, titrating from high to low, you will have the 3 titrations quite similar. But if you titrate from low to high, you will have the first titration different from the other 2. In the case of fast UV, which is the quicker technique, will show more information, as maybe you will see how the decomposition progress. So how you can go from the first titration and to the second, and how the decomposition is worse and worse. So this kind of investigation will be the solution. And actually thinking about it, we had a case in our lab where the sample decomposed actually in both extremes, in acidic and basic media. So for this sample, what we had to do is actually what I explained in my presentation, which is starting at different pHs at different and opposite directions to find the pH which finally the sample didn't decompose. Hope it helps.
Okay, thank you. Let's see. Yeah, we still have time for one more question. Again, if we didn't get to your question, we will offline. Next question. I have a light sensitive sample, so I cannot use the spectrometric techniques, but the pKa is also predicted to be very low, likely outside of the range of the potentiometric technique. What can I do?
Sorry, could you repeat again? A sample that is light sensitive and
Where they cannot use the spectrometric technique.
Oh,
but
got it.
pKa is also predicted to be very low, likely outside of the range of the potentiometric technique.
Okay.
What can I do?
So yes, we have some cases like this one in our lab actually, where we have, yes, a sample is light sensitive, and then even if it's light sensitive, I will recommend you to use the fast UV and the UV metric assays, because at the end, it depends how quick is going to be this decomposition. So if the decomposition is slow, you will have more chance to obtain a result if you have to obtain a result than if you have a quick decomposition. So yes, I recommend to perform the titrations low to high and high to low. Yes, because maybe the acidic or the basic media is going to help to decompose to this light sensitive decomposition. So even that is going to be light decomposition, try to do both media to just confirm that the media is not helping to the decomposition. And actually, I didn't mention in the video, but in the instrument has in the titration and the reference positions like a little black jacket, which you can turn to avoid the light from the room. And so to avoid actually that this light could affect the experiment. So thank you.
Great, thank you so much. Well, that's all the time we have today, but thank you again to our speaker for her contributions today. And thank you all for joining us today and learning about the T3. We want to make sure that we answer your questions. Again, we will do so offline. Be sure to check Pion's website for upcoming webinars. And I'm Marisa Fraser Moreira for Pion. And thanks again for joining us today. And we'll see you next time. Bye now.
Yeah, thank you. Thank you so much.
Bye now.